Welcome to our presentation. This is a major qualifying project on tissue type recognition for laser-based cancer treatments. I am Irene, and these are my teammates, Yixue, Floris, and Kayla. The advisors for this project are Professor Arnold and Professor Fichera. This is a multidisciplinary project such that it involves the application of mathematical sciences, robotics, and computer science techniques.
The target of this project is the in-office laryngeal laser procedure, which is intended to remove pathologies located on the larynx or the voice box. These pathologies, such as papillomas or polyps, can cause speech impairment disorders such as dysphonia. This procedure is performed in a doctor's office as opposed to in a surgical operating room. And this presents numerous benefits. It saves time and money, and the patient does not have to undergo general anesthesia. However, there are some limitations of this procedure. Surgeons have limited control over the laser, and they have to ensure the success of the laser by maintaining a careful balance between overexposure of the tissue, which can cause damage to surrounding healthy tissue, and insufficient treatment of the target tissue, which would result in the need for retreatment. On average, individuals need 2 or more laser treatments due to the inefficient initial treatment of the pathology. A majority of complications arise from surgeons having no assistive tools to judge the effect of the laser on the tissue.
The goal of this research is to tackle these limitations. Specifically, we want to create a tissue classifier to provide the surgeons with additional information about the tissue impacted by the surgical laser, allowing the surgeons to better determine which tissue has been sufficiently treated. By relaying this information to the surgeon, they can better judge the completion of the procedure, reducing the need for retreatment and minimizing complications.
Our approach to this problem contains 4 parts: temperature collection, parameter estimation, classification, and feedback to the surgeon. The temperature collection module in the real-world scenario collects sequential tissue surface temperature data with a FLIR camera. Then the temperature data are passed to the estimation framework, where we use the nonlinear filtering and augmented ensemble common filter to estimate the time-varying parameters of the temperature data. The time-varying parameters can provide an approximation of the tissue properties. Then the estimated parameters are passed to the classifier, which uses label machine learning models to distinguish if the tissue is sufficiently treated. Finally, the state will be returned back to the surgeon to help judge the completion of the treatment. To prove that this approach can work, we have verified it with simulated temperature data, which means that this project is still in early stage. The future steps of this project will require the verification with experimental temperature data.
To validate the approach we discussed earlier, our initial step is to build a mathematical model. We hypothesize that various tissue types have different properties and thermal responses, so we can predict the tissue type by plugging the temperature data into a mathematical model and studying the tissue properties. Figure 1 displays a temperature profile of a tissue surface a second after a laser exposure. We can see that it follows the shape of a Gaussian function. This is one of the reasons why we decided to build a Gaussian model. There are also some assumptions for the Gaussian model, such that the tissue is homogeneous, the surface of the tissue is flat, and that the thermal camera is perpendicular to a tissue. The parameters of the Gaussian model include the amplitude A, standard deviation sigma, and the center location mu. However, we want to assume that the center of the Gaussian is constant. Then we can define the amplitude and standard deviation as time varying. We hypothesize that the change in time varying parameters will assist us in approximating the tissue properties and help us in determining the different types of tissue. Our next step is to generate sets of simulated data to verify our estimation framework, which will be discussed later. Assuming that the tissue has even properties over its surface, we can define sigma x and sigma y to be equal. Then we can define appropriate A and sigma functions in terms of time t to present the change in the temperature of the tissue. To achieve this, we refer to figure 2 that displays the behavior of both A and sigma during a laser tissue experiment from a previous study. From this figure, we can use piecewise functions to present the evolution of time-varying parameters. Figures 3 and 4 are the A and sigma functions we use to generate a set of simulated data. We can see that these functions share similar behaviors as the parameters shown in figure 2. This is a video of a set of simulated data resulting from the previously defined A and sigma functions. This displays a generated laser tissue interaction.
With the simulated temperature data, we want to estimate the temperature parameters, including the amplitude and standard deviation sigma in terms of time. With Gaussian model and the parameters, we can predict the temperature states, which is called model states here. The simulated temperature data we use as the input is called observed temperature data here. With the Bayesian framework, we treat the model parameters and model states as random variables with probability distribution. The joint posterior density, which defines the density of states and parameters given the observed temperature, is proportional to the product of likelihood function and the per hour distribution, where the likelihood function indicates how likely the temperature data are observed if we know the states and parameters, and the per hour distribution encodes no information on the states and parameters before we take the temperature data into account. At each time step, firstly, we predict the states and parameters at current time. And secondly, we correct our prediction with observed temperature. And we repeat this scheme for the entire observed temperature sequence. This figure shows the example estimated parameters of the simulated data. The estimate in red is the mean of the posterior distribution at each time step.
A linear regression model was used to classify different tissue types based on the estimated parameters from the ensemble Kalman filter. We created simulated surface temperature data points with variations in amplitude, and we passed the simulated data through our estimation framework to obtain specific parameters of the data, such as the amplitude and standard deviation. We use the estimated parameters to create predictors in the linear classification model to properly detect the generated type of data. Examples of predictors used were the maximum of the estimated amplitude, the maximum of the estimated standard deviation, and then the mean of both as well. The idea behind classifying the tissue is to bring this information forward to the surgeon to better determine whether the tissue is successfully treated based on its thermal response. You can see two different types of generated parameters on the right, in the figure on the right, and two separate clusters, which allows us to classify between them using a linear model. To conclude, the accuracy of classification is very high for simulated data types with a significant difference in amplitude. A difference of 50 degrees between simulated data types will result in a classification having around a 99% accuracy in the prediction of that type. The accuracy of the classification decreases as the type of simulated data has a similar thermal reaction to the other type, and as the observed data has an increase in noise. To apply this classifier to the surgical environment, we would need to verify our conclusions with experimental data as opposed to the simulated data that we've been using.
Originally, our next step after the validation of the estimation framework with simulated data was to collect experimental data using the setup shown here. Although we collected some data, as a result of COVID-19, further testing was halted and we were unable to complete the validation of our work. In the future, work can be done to use the same setup and create a more documented process for collecting more experimental data for verification. To improve the application of this idea, there's a need for a surgical UI, use of more realistic tissue samples, and use of the robotic arm for automation of data collection. This concludes our presentation. We would be happy to answer any questions you may have, so feel free to contact us via email. Thank you for listening to the presentation, and we hope you enjoyed it.